Thank you, Vanessa. I think I'm in the right channel. Let me see. Yes, I am. Okay. Yeah, so it is, it is my great pleasure to introduce Luisa Iruela-Arispe. She is the chair of the department to which the Center for Advanced Microscopy belongs, so that's how I know her. She has a fascinating career in something I find extremely exciting, which is the blood vasculature. And in the context of how I got to know her, so I had read her papers, I knew about her. And when I first interviewed at Northwestern University, I interviewed with her. And I noticed that there was a recognition at the back of her office. So more or less, I imagine she's facing it right now, which was for Best Mentor. I think she's won several awards of that. And that was very, I noticed it. And to me, it's something very important. So to me, it means that the person cares about the people she's training. And so the more that I've been here, a member of this department, the more people have shared the changes that she has brought about as chair in terms of DEI. I believe this is something that she values, and it permeates throughout the department. She has fostered the inclusion of more women, of young faculty, and of people of different races. And she has been very supportive of initiatives we have had in terms of neurodivergence too. So all in all, I think she's recognized in the department as someone who has fostered a huge amount of positive change. So it is with great pleasure that I'm welcoming her to this speaker series for Hispanic Heritage Month. I was going to give a brief overview of her career, but I think she will do that. So I will, with this, leave the floor to Luisa.
Thank you, Mariana, and thank you all for being here. It really is my pleasure to be part of this series. It's actually an honor for many reasons. And obviously my heritage is also Hispanic, and I am very proud of being part of this heritage and be a force for change in science. So I will start by sharing my screen. And I hope you can all see it. So the title of my presentation is Endothelial Macrophage Interactions in the Vascular Wall. And I will try to intertwine this presentation with several aspects that I find that might be appealing to you. But along the lines of what Mariana was saying, the first thing is to say that I was born in Spain. I'd immigrated from Spain to Argentina at the age of 5, obviously following my parents that were interested in relocating because of political reasons. At that time, Franco was the president, and the dictatorship did not agree very well with my father, who decided to just immigrate. And so we did so. I was the only child, remained the only child for a long time. And I did a lot of my education in Argentina until I was 13 years old, at which time problems in Argentina then made my parents move to Brazil. And so here I am in a very different country with a different language, and trying to, as an immigrant, to learn and continue my education. So in Brazil, I did my high school, and then I followed university. I did biology, started my master's degree, finished my master's degree, got a job at the State University of Rio de Janeiro. But I wanted to study endothelial cells. I was very stubborn about that. And the program that I was at the USP did not have endothelial cell biology. And so I decided to pursue my dreams and do my thesis in Seattle at the University of Washington. And so at 23, very unusual for a young Spanish background woman leaving their household and leaving alone in another country like the United States, very far away from my parents. I did so to really pursue my dreams, and it was very lonely. And I'm sure that as some of you who have relocated to other countries can sympathize with that. But it was all very, very exciting because the world of science was just marvelous to me. And so I spent several years in Seattle, and from there, I finished my PhD, I finished my postdoc, and decided to actually stay in the United States to pursue my academic career. This was a time of a lot of uncertainties and a lot of difficulties, but I was able to get my first faculty appointment at Harvard Medical School and in Boston. And so I relocated to Boston at the age of 29, and I started essentially my independent career as an investigator. From there, I married my husband, who is American, but also was born in Spain. It's a complicated story. But in any case, he was a postdoc at that point. We wanted to both have jobs, and he found a job at UCLA. And so I relocated to Los Angeles at UCLA and had my second faculty appointment at the age of 33 in a different institution, continuing our research on blood vessels. And then from there, moved to Chicago, where I am today. And this is my third faculty appointment, and I'm chair of the department, continuing to work on blood vessels and being very grateful to be in a really terrific institution in trying to build a very strong department. So all of this has happened as I study blood vessels. And so the story that I will tell you today has to do with a new cell type, very exciting, that we discovered as we were interested in studying the aorta. And so this is the picture of the largest vessel that we have in our body, the aorta, that emanates from the heart, and it goes all the way in the back of, attached to this spinal cord, actually. And this blood vessel, if we cut transversely, shows multiple layers of cells. The innermost layer is called the endothelium, is also collectively termed tunica intima because it includes other layers, and because it's the innermost part of the vessel, that is tunica intima. The intermediate one is the tunica media, which houses the smooth muscle cells, and this tunica is responsible for the contractility of the vessel. The outer layer is the tunica adventitia, and this external layer houses a lot of connective tissue and fibroblasts. So our focus has been on the endothelium, particularly this single layer of cells that faces the blood and is responsible for much more of the communication between the blood and all the organs in the body. The endothelial cells are responsible for many functions. One is barrier. So without the endothelium, the blood will be extravasating and getting out of the circulation. Not a good thing. It's also responsible for immune trafficking. So if you will, the endothelium is the door that needs to be open for leukocytes and other cells of the immune system to extravasate and initiate any type of immune response. So the endothelium has to give permission to those cells to traffic. And many other functions, and one of them is actually to prevent clotting. It will not be a good thing if clots happen inside of the blood vessels. And when they do happen for pathological reasons, they result in stroke or heart attacks. And so this is a very important function of the endothelium. And what I will tell you today also relates to this function. The interesting thing about the endothelium is that, again, it really is a very active layer of cells that is interacting with all these leukocytes and all the blood cells in the body. And so usually this interaction tends to be transient. And so that cells will adhere, they will roll, form firm adhesions, and then transmigrate through diapedesis in a manner that now enables them to interact with any setting of inflammation and act upon them. There is another setting in chronic atherosclerosis when there is a very active interaction of these immune cells. And they tend to stay a little longer, but when they stay also underneath the endothelium. So the entire field of vascular biology has this notion or this dogma that interactions of the endothelium with inflammatory cells are transient. They are not, in that transient interaction occurs either during acute inflammatory states or even in chronic inflammatory states. They never stay in the endothelium, they go through the endothelium. I'll show you today that that is not the case. This dogma is not particularly true. And so here enters a fantastic graduate student, also Latina, Gloria Hernandez. She was a graduate student in my lab. Now she's working at Genentech in the industry. And she took on this project. It was mostly because of morphological observation in the beginning. I told you that the interaction with inflammatory cells is transient, and this is the dogma. But when Gloria was looking at the aorta, and here she actually opened up the tube and flattened it open to look at it on the confocal microscope, she found that if she stained for inflammatory cells in green, all those inflammatory cells decorated the innermost curve of the aorta and also the branches areas. And this happened in a very healthy animal and relatively young animal. So she was puzzled by the presence of these inflammatory cells because this is not what it's supposed to be. This is not what we learn in school. So how can we explain the presence of these cells in the context of a normal situation? And they were also beautiful. And so she was very attracted to how spindly they were and how they were interacting with the endothelial cells. At this point, she didn't know whether or not these interactions were underneath or if they were on the surface. And so she devised very interesting techniques with the help of Cam and a lot of support from our colleagues here at Northwestern. And she found that, in fact, these cells were on the surface, but some of the processes went underneath the endothelium, which is now here in red. So I always show endothelium in red and the immune cells in green for most of the talk. And so this immune cells were being essentially saw inside of the endothelium. And this was, again, in normal situations. This is another approach that she used by visualizing the cells. So it's always important to use different types of approaches. And she found that these cells were, again, extending their processes underneath the endothelial cells. And sometimes we will see the little process on the top appearing on the other side. So they were really embedded in this layer of endothelial cells. Interestingly, she found that these endothelial cells were particularly associated with areas of flow that was disturbed. And so this hemodynamic forces can be either linear, like what we see here, the arrows, or they can be disturbed. And so every time that there was a branch, we will observe the presence of these immune cells. And it's been known for a long time that areas that are laminar flow are protected, and areas that are disturbed are atheroprone. And so atherosclerosis develops in these regions. So she went on to try to understand if, in fact, these areas of disturbed flow were decorated with these immune cells. And so you can see here an opening inside of the aorta. These are the intercostals. We don't see immune cells at this point at 1 week, but very quickly after time, they start populating areas of openings. And again, in higher magnification, they're also present in the anterior aspects of any large branch. So you can see them here interacting with the endothelial cells that are in white in this case. So she became very interested at understanding the relationship between these branch openings and these immune cells, and found that these immune cells were associated with areas of deposition of this protein called fibrin. Fibrin or fibrinogen circulates in the blood, and it's not very good to have a lot of fibrin inside of the blood vessels, because fibrin can actually form a mesh and initiate the process of clotting. So it was peculiar that we found the association of these immune cells with fibrin. It seems like they were binding to fibrin. What she found is that these areas of fibrin were present in the thick regions in the aorta and also in the intercostals. And with age, there was more accumulation even in the linear regions of the aorta with linear flow. So let me first now kind of sort of give you a sense of the first set of conclusions. So I'll have like 4 parts to this talk. So I hope I show you evidence through the beautiful imaging that Gloria did, that in healthy adult mice, there is a concentration of immune cells that can be identified by this very important marker, CD45. And these regions are associated with disturbed flow, like in branch openings and the lesser curvature of the aorta. And these intimal cells, immune cells, progressively increase with time, but they're not associated with atherosclerosis or regions of hypercholesterolemia. This is a normal state. We also see it in humans. The cells also are embedded in the endothelium. They are being saw in the cells. So now Gloria was interested in trying to understand further, what are these immune cells? There are many types of immune cells, so we need to identify the identity, the cellular identity of these immune cells. We wanted to understand where they're coming from, and we wanted to understand how relevant are they for the biology of blood vessels. So to get at the first issue, what she did was a technique that now is rather common. She did a single-cell RNA-seq experiment, and for that, she isolated young mice and old mice. She collected the arch, and she collected the descending aorta, and she peeled off the cells to identify them transcriptionally through this technique of single-cell RNA-seq. She then obtained the results, and what we found, very surprisingly, is that the population of cells that were associated with the aorta were actually macrophages. And this was found because of the markers of these macrophages. Macrophages usually have expression of CXCR3 CX3CR1, and they also express CD11 at times. They also express LIVE1 at times. And so the interesting thing is that, so they were definitely macrophages, but they were different from the precursors of the macrophages that are circulating, which are called monocytes, a type of leukocyte. They were also different from the macrophages that are located in the tunica adventitia, which is the outermost layer that I mentioned to you in the very beginning of the talk. And so even though the adventitia macrophages and the cells that she found were actually derived from monocytes, as you can see here, they're very different transcriptionally. And we can know that because everything that is yellow shows a very high expression of a particular subset of genes. So these are the transcripts for these genes. And we can see that the monocytes express mostly a cohort of genes that are then silenced in the adventitia macrophages and in what we call now macrophages. And so we call them macrophages because they are aortic intima resident macrophages. So they are macrophages, but they are inside of the vessel. They're not outside in the adventitia. And so Gloria related these macrophages to essentially the computer that we all use, right? So, and they have very distinct signatures. And so here we are comparing through violin plots the expression of specific genes in monocytes, adventitia, macrophages, and the macrophages. And as you can see, there is a very distinct pattern of expression that is unique. To validate that this is really the case, what Gloria did was to perform immunohistochemistry. And so she showed that the genes that were expressed in the macrophages by single-cell RNA-seq were also expressing, now in red, this marker. So CXCL16 is a unique marker for macrophages, as well as MMP13, a matrix metalloprotease, and CD11C, that normally is associated with another inflammatory cell type called the dendritic cell, but in this case, it's also shared by the macrophages. So this is a staining in the intima, that is in the inner layer of the blood vessels. When she looked in the adventitia, she found that none of these markers that were present in the macrophages were present in adventitia macrophages. And so these markers are really unique to this population of cells that is very unusually located in the intima aspect of the blood vessels, of large blood vessels, in areas of disturbed flow. So essentially, intima immune cells are found in the aortic arch and in the age-descending aorta. And they're a unique population of macrophages that we call them aortic intima resident macrophages, or macrophages. So now, so this is what she completed this goal, which was to identify the transcriptional and cellular identity of these immune cells. So where do they come from? And this is an important question because immune cells can be originated by from many different sources. And depending on their source, they can be temporarily or more permanent, and they can have different potential for differentiation. So this was a very important question that she wanted to answer. So when these intima resident macrophages first appear in the aorta. So Gloria went back and tried to look at how early. I mean, we knew that by 8 weeks they are there, but what is the earliest time that we now don't see them? And she had to go all the way to embryonic stages. And this is an aorta from an embryo at embryonic day 18.5. This is very difficult to do because the animal is very, very small. And she's very talented at opening this aorta. And this is actually the aorta. And you might say, well, what is this very big vessel that you see right next to the aorta, which is located here on this whole mount image? So this large vessel is called the ductus arteriosus, and is very open here because during fetal life, the ductus arteriosus is the region that enables the communication or the open circulation with the lung. And I'll show you this more in a minute. So by embryonic 18.5, and the mouse embryo, I should tell you, is born at about 20 days. So right before birth, they're not there. The first time we see them is in postnatal day 1. So one day after these animals are born, they appear, and they start appearing, guess what? Now in the ductus arteriosus, that is contracting. It's very, very important to contract this vessel, because if it doesn't contract, then we will have what is called blue babies, where the circulation of the heart and the lung are shared, and so this blood is not oxygenated properly. And so you can see here in the schema how this happens. So the ductus arteriosus communicates very openly the circulation of the lung with the systemic circulation. And it's very important for this to close so that the venous circulation does not mix with the arterial circulation. And so, and over time, this is completely blocked, and now the two circulations are separate. And so this is where this cells originate. And in fact, it's very interesting to see that the endothelial cells change over time, and they become a little bit more polygonal, while actually in the descending aorta become more elongated. So this is where the macrophages are found. And we can see that these macrophages are emanating, they come out of this ductus arteriosus as the ductus is closing. So we can see them here kind of migrating like ants coming out of the hole. And in fact, they are sharing some of the markers that we identified in the single-cell RNA-seq of the adult, meaning these cells are the very same cells that we identified in the adult, or at least this is one piece of evidence that that is the case. They also express, if you remember, CXCL16, which is one of such markers, and they overlay with CD45, and they're very, very dendritic. They have a lot of processes. So we really believe that the same cells that we saw in the adult really are the descendants of this particular cells coming out from the ductus arteriosus. So second set of conclusions is that the first time that we see MACARES is really postnatally. I didn't show you the data that tells us this, but they derive from definitive hematopoietic progenitors that seed the aorta from the ductus arteriosus, and they migrate in areas of disturbed flow. Okay, so just to be sure, are the adult intima resident macrophages truly originating from that ductus arteriosus? And are they self-replicated? Do they divide? Or are they just, they're coming from the circulation and replenished? So we use some interesting genetic techniques that allow us to essentially label these cells, only these cells, in 3 different colors randomly. So some groups of cells will be cerulean, others orange, and others cherry, using this genetic strategy. And so we did this very early during development, and if the cells were really descending from the initial cells when they were seeded at these early time points, we should see them very later on in the adult. So when we harvest the adult after induction of the system, what we found is that indeed we could identify clones of different colors seated in the aorta. So this type of experiment indicated to us with certainty that those macrophages that were initially coming out from the ductus arteriosus were indeed the same ones, or the precursors of the ones that we saw in the adult. So the Maccaires, in fact, originate. We did, again, fight mapping at different times, and we did immunohistochemistry with CD45, and we can see that they perfectly overlay with each other. So this is the appropriate technique to visualize and to ask the question of where they're coming from. So essentially, the Maccaires are a unique population of macrophages. They are developmentally programmed. They are not inflammatory. They have nothing to do with inflammation. They're just part of the blood vessel at that time during development. And then they stay regardless of any particular disease. They stay in the lumen of the blood vessel. Until the age they actually expand over time. Okay, so no Maccaires during development, and then they come at P1 and P5, and they extend, they express particular subset of markers, and they are mostly located in areas associated with turbulent flow, and then they expand with age. Okay. So finally, the last thing is, what is the biological relevance? And to understand this was like a serendipitous finding that Gloria stained for fibrinogen. And what she found, remember that fibrinogen was associated with areas of turbulent flow. She found that the Maccaires were actually very much link to the presence of fibrinogen. So fibrinogen here is in white, and you can see with CD45 the presence of these macrophages, and then the endothelial cells in red. So in fact, these macrophages are decorated with fibrinogen. So they are covered with fibrinogen. So the macrophages here are in this color pink, and then they are surrounded by fibrinogen, as you can see in red. So in fact, these macrophages are digesting the fibrinogen, which is very important to remove. Otherwise, clots will happen in the aorta. So to understand whether or not this was, in fact, their function, what Gloria did was to remove these cells from the adult mice by using a genetic trick. And the trick was to actually express diphtheria toxin on the surface of these macrophages only. And then by adding diphtheria toxin, that will trigger cell death. And so we can verify that this worked, because if we stain the aorta with the CD45 and then we add diphtheria toxin, after 1 day, they're almost completely gone. So in fact, this is what happened. It worked. And so what happens to these mice when we remove them? Or what happens to the cells? So the first thing we see is that in the absence of these macrophages, the endothelium looks different. The endothelial cells actually look smaller, and they're actually contracting. And we know that they're contracting because they're expressing phospho-MLC, which is a marker for contraction of the endothelial cells. So in a normal control mouse, phospho-MLC is present in the macrophages, but when the macrophages are gone, now is the endothelial cells. They're very actively contracting. And so we could see that essentially when we deplete the macrophages, what is increasing is the amount of fibrinogen. So in green here now we're seeing accumulations of fibrinogen. And as I mentioned to you, fibrinogen is not very good to have inside of the vessel because that can result in the formation of clots. And in fact, we can see that in a control, the amount of fibrinogen is relatively small, but in the absence of these macrophages, there is an accumulation of fibrinogen, and there are fibrils that form meshes, and those meshes can now trap red blood cells and form microclots. Those microclots are not good to have in the circulation, and we can see here one big clot of fibrinogen. There is a rupture in the endothelium of the aorta, lots of microclots inside of here, and the animals start dying. And so what happens here is that, and there is a readout for intracoagulation, which is the dimer that reveals this very clearly. And we can see that in multiple organs, there's clotting of blood vessels. And so the animals, a lot of the animals die, 50% of the animals die without these macrophages. And so this kind of sort of finishes the presentation. So the working model that we have based on all the work that Gloria did was that essentially the endothelial cells are interacting with these macrophages. And these macrophages are really, really important at making sure that the fibrinogen that is formed because of this turbulent flow, this disturbed flow in areas where there are branches, is degraded by the fibrinogen. And that is happening because they make a lot of plasmin, and plasmin cups up the fibrinogen. Now, when the macrophages are gone, that cannot happen. And so the fibrinogen forms fibrils that then now traps red blood cells, clots are formed, and the animals suffer from those clots. So with that, I just would like to finish the presentation and really thank Gloria, who did all of this work. It was very beautifully published in a high reputable journal, Nature Cardiovascular Research. And this was done, actually, a big part was during the pandemic. And you can see everybody here cheering Gloria during the pandemic. And we cannot do this in silo. We really owe to a lot of our collaborators within the lab and outside the lab in multiple institutions. And so with that, I just finish, and I'll be very happy to answer questions.
Thanks a lot, Luisa. That was a fantastic talk. So many messages. Does anybody have a question from the audience?
I'll stop sharing.
Yeah, you can ask us in the chat. Otherwise, I can, if I can start, I can start with some questions, and then people can gather the courage to ask. So about your career, so some of the things that you showed were to some extent paradigm shifting, or paradigm shifting. How difficult is it to do that and to defend that and to the whole credibility? Also, you were very young in your different positions. Was that challenging?
Yeah, so lots of questions there. Yeah, I mean, I think the most challenging position was the one that I had in Brazil, because I was very young. I was 23, and I actually was teaching to medical students histology and embryology, as well as to biology students and pharmacy students. And they were, 2 years younger or so than me, or 3 years younger. So, and this happened because I skipped a few grades when I moved from Argentina to Brazil. So that was very challenging. that was hard, and particularly being a woman at that time, it was very difficult. But I tried to focus on the job that I was doing and not pay attention or get distracted or get derailed by comments or any issues. But yeah, that was hard. In terms of fighting dogma, yeah, I is very difficult, right? So these ideas are instilled in the literature, and they're instilled for a particular reason. People observe this, they make these observations, and everybody agrees and believes it. And then you find something that doesn't fit. And then you need to essentially prove again and again and again. So it took a long time for us to actually get this publication across because of this defiance of what it was believed to be true and cemented. And we needed to break that cement and say, well, let's be comfortable with being uncomfortable. This is different, and we need to understand it. So that's the approach.
That's very interesting. Piyush is asking, is it a parsimonious model where more turbulent blood flow leads to more macrophages throughout the body?
Yeah, it is. It's every time that you have turbulent flow, you will see these macrophages. So now the reason why people didn't see this before, people have been looking at blood vessels for a very long time. And the reason is actually the technique that the Gloria really utilized. She opened it up as a sheet. So the macrophages have these processes, and these very long processes. And the processes, when you cut it, it will look like a little green dot, and you're not going to give any significance to any green dot. You're also not going to give significance to an inflammatory cells on the surface of the endothelium because you will think that is going through. So the only time that, allow us to pay more attention is when she opened it up. And then when she opened it up, she saw these beautiful cells that were never described before. And that is what really led us to, continue this particular research. And so in human, unless you actually expose the endothelium in that manner, you're not going to see them. So it's the technique that allows us to see it.
What do you think? So while there are more questions, do you know if there is, so in other places where there's an accumulation, not exactly like what you found, but in general, it includes places of macrophages. I mean, it's areas of surveillance. Do you know if there's a need also for that in these particular regions? Increased surveillance of anything, pathogens or anything?
Yeah, I mean, I think that there is also the possibility that these cells are involved in uptake of viruses and kind of preventing. So they are right there, right, as the blood is coming out of the heart. And that blood is going to be now disseminated to the rest of the body. So I think it could be a very opportunistic way of just like, okay, let's filter anything that is bad coming out of here. And obviously there are other colleagues that have been now, investigating and follow up this. And there is this tendency to believe that they are also there to, take out bacteria, viruses, and so forth and so on. So that's a very, appropriate, notion.
Mm-hmm. Thanks. Andres Gutierrez is asking, hi, Luisa, awesome talk. Have you seen this macrophages specifically in other locations?
Yeah, to that, that discussion. So definitely they are located in areas of turbulent flow. Now, in terms of what about other organs in other regions that are not associated with the circulation, and that is not necessarily the case. The only time when we saw some association with some macrophages was similar, but not the same identity, but some similarities were in the lung. So there is a population of resident macrophages in the lung that were published by others. And we found by comparing the single-cell RNA-seq that there was a significant overlap, not entirely identical, but more similar than the adventitial macrophages, for example. Great question. Great questions.
Piyush is, thanks, Andres. Piyush is asking another question, which is very fascinating. As a biophysicist, I wonder if there is a cutoff Reynolds number around which these markers get recruited.
That's such a great question. And so we're actually, we're teaming up with biophysicists, and here in Northwestern, there's a fantastic group that is very interested in really quantifying flow in different regions and characterizing it in detail. And so we're very interested in this very same question that you asked. And so we are now initiating experiments with them to try to see if we can actually link these numbers and also disturb the flow in different ways. So there are circumstances where the ductus arteriosus is not closed, right? It remains slightly open. So that affects also the level of turbulence that is happening, the biophysical properties of that flow. And we wanted to see whether that is retained. So it will be really fascinating to know if the numbers that we obtain in a wild type situation with the ductus closed are different or similar than in situations where the ductus is open. Awesome question.
While there are other typed questions. Okay, he says, thank you. I have a question, which I think is relevant to several people in the audience that I know, which is, you had a great mobility during your career. What would be the most important advice you give to someone? You touched on one thing, which was how lonely it can get, but you overcame it and you're very successful. So what would be your main advice on mobility?
I think, yeah, it's hard, right? Because I think that just like many Hispanics and being a woman and our parents are very very holding on to the children is very difficult to let go, and so that link is hard. But for me, I did not think that I was going to come to the United States to actually live here. And my mother frequently reminds me, well, you told me it was going to be just for a few years. And it wasn't. And partially that's because, I met my husband, obviously, and I ended up staying. But I think you have to be fearless, particularly when you're young. And, the world is such a fantastic place to explore. And if you have the opportunity, take it. That's my advice. Obviously, it's very personal and it's something that you need to feel comfortable with, leaving your family. And this is very personal. But I just had the great mobility because I just wanted to do stuff. I wanted to really pursue my dreams, and I did not want fear to interfere with my goals. So I just tried to ignore it. I don't know what's happening. Never get phone calls, and now I'm getting all of them. Apologies for me. Okay.
So if anyone is interested, I think you gave an interview to, what was it, Nature something?
Oh yeah, Nature Cardiovascular Research, actually, where we published this work, and I kind of, they were asking us. This was the first year that the journal came about, and they asked the senior authors of all the papers that were published that were women, just for the International Women's Day, I think, to talk about their journeys and why they became scientists and things of that sort. So yeah, there's a little article there.
Okay, so if there are no more questions, unless there are, Thank you so much, Luisa. I mean, all the presentation will be available in the YouTube channel of BINA. Often people go on and watch it afterwards, whoever wasn't able to join. And then if you're happy with it, then there's been also contacts with the speakers. So if they're interested in following up, so that would be, if you're interested.
By all means, you can all send me emails if you have any additional questions or you know, anything that I can help you with. I'll be very happy to reply to any of the emails from here. And thank you, BINA, for doing all of this. I mean, I think the organization is fantastic. I'm really delighted to be part of it. And I am very appreciative to have this. I wish I had it when I was young, but I'm really grateful to all of you for all the time and effort that you put into this.